Hello everyone, welcome to my channel. Myself Dr. N.L. Terdal, working as an assistant professor in the Department of Chemistry, JSS Arts, Science and Commerce College, Gokak. The college is affiliated to Rani Chennamma University, Belagavi. So the today's topic of discussion is the continuation of electrogravimetry. So in the previous videos we have discussed about introduction to gravimetry, classification of gravimetry. In the classification we have seen in detail studies about electrogravimetry, which is designated as EEG, and then we have discussed about basics and principles of EEG. In the today's discussion, we will see, or we are going to see the instrumentation of electrogravimetry. So before going to this instrumentation, we will just recap the principle of the electrogravimetry. So electrogravimetry is a method in which the analyte, usually the metal-based analyte, is being determined through the electrolytic deposition onto a suitable electrode, which is the suitable electrode is known as working electrode, under controlled experimental conditions. So under controlled experimental conditions, electrolytic deposition is done by by using the suitable electrode or working electrode. So in electrogravimetric method, the filtration is not required as we are doing electrolytic deposition or electrodeposition, and under optimum parameters, under controlled conditions, the co-deposition of two metals can be often be avoided in electrogravimetric technique. So it is a simply analytical technique where we can determine the metal based analytes by electrolytic deposition or electrodeposition. So how we are going to explain this electrogravimetric principle? It is simply based on two laws. First law is Ohm's law. As we know that I is directly proportional to V by R, that is directly proportional to voltage and inversely proportional to resistance. So that is Ohm's law, and it is also based on the Faraday's laws of electrolysis. There are two laws. One law, Faraday's first law, and Faraday's second law. So that we have already discussed in the last in the previous videos. So in this video, we are going to discuss in detail about the instrumentation of electrogravimetry. There are two types of electrogravimetric methods. So based on the two entities, one entity is current and other entity is potential. So if current is constant, and that type of the electrogravimetric method is constant current electrolysis, and the second method is constant potential electrolysis. So these are the two important methods in the electrogravimetric methods. So we shall discuss one by one here. The first one is a constant current electrolysis. This is a very important method. People are using this method in the research studies as well as academic studies. So here is a apparatus. You can see it is very clear that we have a very nice apparatus here. In this apparatus, we have what we call it is ammeter, which is used to measure the current. Ammeter, we know well. So this is a voltmeter. We can also call it as a potentiometer. So where we can measure the voltage of the circuit, and this is the battery. The battery used in this electrolysis setup is, that is constant current electrolysis setup, is around 6 volt. So this is the circuit, external circuit. We have two electrodes here. One is you can see this is a blue platinum gauge, which is acting as a cathode. So one more, there is a spiral platinum electrode, which is acting as a spiral platinum electrode is anode. So then this is a magnetic stir, which is needed to homogenize the solution throughout the electrolytic process. So this is the unlight solution, you can see, and this is electrolytic cell, we can say. So this is the parts of the, or we can say apparatus, which are involved in the constant current electrolysis setup. Okay, let us see what is the construction here. The constant current electrolysis setup is constructed by referring this figure, or we can say constructor is as shown in the figure. Working electrode, that is platinum gauge cathode, So which is a circular electrode on which the analyte metal is going to deposit under applied potential at constant current. Under applied potential at a constant current. So this is the principle of the this is the function of the platinum gauge, that is a cathode, where the analyte metal is going to deposit on the surface of the platinum gauge cathode. Well, so what is the function of this platinum spiral anode? So this is acting as an anode, so which can complete the circuit and gives us the current as well as voltmeter readings. So then magnetic stirrer is really required to homogenize the solution of the analyte during the electrolysis setup. So this is the construction. It's a very simple. So here we are just applying the potential at constant current, and then we are depositing the unlight material on the surface of the platinum gauge cathode. And another electrode is anode, of course. So we have two electrode system here, and then we are measuring the we are just applying the potential by using potentiometer here, and then we are measuring the current, that is the by application of the constant current. So we have construction here, and then let us see the what is the working. So how it is going to work. The electrode deposition or the electrolysis of metal based on light is carried out by keeping the current constant. So that I already said you, the periodic increase in the applied potential is required by adjusting the potential of the cell as the electrolysis proceeds. So it's very important. The periodic increase in the applied potential is required as electrolysis proceeds. So the apparatus for constant current electrolysis consisting of suitable cell and a direct source. So around 6 volt source is used. An ammeter and voltmeter are used to indicate the current and applied voltage respectively. Electrolysis starts when voltage is applied at constant current, and it continues until the complete deposition of analyte onto the cathode. So once we deposit all the analyte which is present in the solution on the surface of anode, then we just take out the anode, that is the platinum gauge, and then wait before electrolysis and after electrolysis. The difference in the weight of the cathode is the weight of the analyte present in the solution. It's very simple. So we will discuss now more on this current electrolysis by taking the simple one example, that is the determination of copper by using this constant current electrolysis method. So we shall see in the later videos. So in the second method is constant potential electrolysis method. So here we are using three electrode system, and in the constant current electrolysis method we are using two electrode system. So here again the same ammeter, voltmeter, battery, working electrode is again the platinum gauge, reference electrode that is standard hydrogen electrode, and third electrode is counter electrode, that is platinum wire, and next is magnetic stirrer electrolysis is the same setup. So here is a thing. So we have a DC power supply here. You can see in the constant potential electrolysis setup. So we have two types of circuit here. One is electrolysis circuit, where you can see we have applying the power. We have DC power supply, and then we have a rheostat here to control the potential, digital current meter, that is ammeter, and here is a, digital voltmeter, that is a reference circuit. So these are connected as shown in this figure. Like working electrode, we have three electrode system here: working electrode, then reference electrode, and then counter electrode. So the function of the working electrode is to deposit the analyte material on the surface of this. So this is usually platinum gauge. And next is a reference electrode. This is very much important to important to apply the potential with reference to this reference electrode, which is standard hydrogen electrode. And next is a counter electrode. We just need to complete the circuit. That's why we are using this counter electrode to measure the current. So this is all required in this constant potential electrolysis setup. So how it is going to construct? It's very simple. Constant potential electrolysis setup is constructed is as shown in the figure. Working electrode, that is platinum wire, platinum gauge, on which analyte metal is going to deposit at constant potential. So this is important, and the reference electrode is required to apply potential with reference to the a reference electrode. Counter electrode is needed to complete the circuit to measure the current. So this is about the construction. So in the working by controlled potential electrolysis, it is possible to separate two metals, or we can say elements in general, whose deposition potential differs sufficiently by a few tenths of a volt. The potential of the cathode is controlled so that it never becomes sufficiently negative to allow the deposition of the next element. So why we are using the reference electrode, why we are using three electrode setup. Here is a gist. The potential of the cathode is controlled so that it never becomes sufficiently negative to allow the deposition of the next element. So if you are not applying the potential with reference to the reference electrode, so what is happening? So that the working electrode potential is sufficiently negative, and then it becomes co-depositing the metals or next element taking place. So in order to avoid such kind of issues, so we are going to introduce three electrode system where we can apply the potential with reference to the reference electrode. So as can be seen from the potential of the cathode becomes negative due to concentration polarization, and that co-deposition of the other species begins, therefore the analyte is completely deposited. So in order to avoid the co-deposition, or in order to avoid the deposition of the next element, so we need to apply the potential very minutely. So we need to have a reference electrode for this. So that's why we are using three electrode system in constant potential electrolysis. So we have discussed two types of electrolysis setup. One is a constant potential electrolysis, and second one is a constant current electrolysis setup. So in the regular lab activities, so we people are using constant current electrolysis setup. We have a specific example that we will see in the next video. So if you have any questions regarding these two electrolysis setups, so please write in the comment box. I will answer you guys. Thank you.